Chapter 8 Neurons, Cellular and Network Properties. This first slide provides an overview of the chapter, outlining the key topics that will be covered. It begins with the organization of the nervous system, explaining the structural and functional divisions, including the central and peripheral nervous systems. Next, it explores the cells of the nervous system, focusing on neurons and glial cells and their roles in supporting neural function. The section on electrical signals in neurons covers how neurons generate and conduct action potentials through ion movement and membrane potential changes. Cell-to-cell communication in the nervous system highlights the mechanisms of synaptic transmission, neurotransmitter release, and signal propagation. Finally, integration of neural information transfer delves into how neurons process and integrate signals through summation, inhibition, and neural pathways to generate appropriate responses. The nervous system is a complex network responsible for transmitting signals throughout the body, allowing communication between different organs and coordinating essential functions. At the core of this system are neurons, specialized cells that process and transmit information using chemical messengers known as neurotransmitters. The nervous system is divided into two main parts: the central nervous system, CNS, and the peripheral nervous system, PNS. The CNS consists of the brain and spinal cord, acting as the command center for the body by processing information and generating responses. The PNS extends beyond the CNS, connecting it to the rest of the body. Within the PNS, sensory or afferent neurons carry information toward the CNS, while efferent neurons transmit signals from the CNS to muscles and glands. The efferent neurons are further classified into the somatic motor division, which controls voluntary movements, and the autonomic division, which regulates involuntary functions such as heart rate and digestion. The autonomic nervous system is divided into two branches: the sympathetic division, which prepares the body for stress and emergencies, and the parasympathetic division, which promotes relaxation and energy conservation. Another important component of the nervous system is the enteric nervous system, ENS, which consists of a network of neurons embedded in the walls of the digestive tract. Although it is influenced by the autonomic nervous system, the ENS is capable of functioning independently, regulating digestion and gut activity without direct input from the brain or spinal cord. Understanding the terminology in neuroscience is essential for clarity and accuracy. Many terms used in neuroscience have synonyms that may appear in different contexts or textbooks. This section highlights some of the key terms and their synonymous counterparts. An action potential, commonly abbreviated as AP, is also referred to as a spike, nerve impulse, or conduction signal, all of which describe the electrical activity of neurons. The autonomic nervous system, responsible for involuntary bodily functions, is also known as the visceral nervous system. The axon, which carries nerve signals away from the cell body, is often called a nerve fiber. Axonal transport, the process of moving materials along an axon, is synonymous with axoplasmic flow. At the end of an axon lies the axon terminal, which can also be referred to as the synaptic knob, synaptic boutton, or presynaptic terminal, all of which describe structures involved in neurotransmitter release. The axoplasm, which refers to the cytoplasm within an axon, plays a crucial role in neuronal function. The cell body, which houses the nucleus and essential organelles, is also known as the cell soma or nerve cell body. Surrounding the axon is the cell membrane of an axon, often called the axolemma. Glial cells, which provide support and protection for neurons, are frequently referred to as neuroglia or simply glia. Similarly, an interneuron, which connects sensory and motor pathways, is sometimes called an association neuron. The rough endoplasmic reticulum in neurons, a key site for protein synthesis, is also known as Nissl substance or Nissl body. Lastly, a sensory neuron, which carries signals from sensory receptors to the central nervous system, is also referred to as an afferent neuron. The nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system, which includes the brain and spinal cord, processes information and coordinates responses. The peripheral nervous system connects the central nervous system to the rest of the body and is divided into sensory neurons, which carry information to the central nervous system, and efferent neurons, which transmit commands from the central nervous system to muscles and glands. The efferent division consists of the somatic motor system, which controls voluntary movements, and the autonomic nervous system, which regulates involuntary functions such as heart rate and digestion. The autonomic nervous system is further divided into the sympathetic division, which prepares the body for action in stressful situations, and the parasympathetic division, which promotes relaxation and digestion. Additionally, the enteric nervous system regulates digestion and can function independently of the central nervous system. These interconnected systems allow the body to process sensory input, integrate information, and generate appropriate responses to maintain balance and function. This concludes our discussion on the organization of the nervous system. Neurons are the fundamental units of the nervous system, responsible for carrying electrical signals that allow communication throughout the body. As the functional units of the nervous system, neurons play a crucial role in processing and transmitting information. Neurons can be classified based on their structure and function. Structural classification is determined by the number of processes extending from the cell body and includes multipolar, pseudo-unipolar, bipolar, and anaxonic neurons. Functional classification is based on their role in the nervous system and includes sensory neurons, motor neurons, and interneurons. Sensory neurons, also known as afferent neurons, transmit signals from sensory receptors to the central nervous system. Motor neurons, or efferent neurons, carry instructions from the central nervous system to muscles and glands. Interneurons act as connectors, facilitating communication between sensory and motor pathways. Each neuron has a distinct structure that supports its function. The cell body contains the nucleus and organelles essential for maintaining cell function. Dendrites receive incoming signals from other neurons, while the axon transmits electrical impulses away from the cell body. Additionally, axon collaterals, axon terminals, and synaptic bulbs are critical for communication between neurons, ensuring the transmission of signals across synapses. Neurons can be categorized based on their function and structure. Functionally, neurons are classified as sensory neurons, interneurons, and efferent neurons. Sensory neurons detect external and internal stimuli, transmitting signals to the central nervous system. Some sensory neurons, such as those for somatic senses, have dendrites and an elongated axon covered by Schwann cells. Others, like neurons responsible for smell and vision, have specialized structures adapted for their specific functions. Interneurons, located within the central nervous system, act as communication links between sensory and motor neurons. These neurons typically have multiple dendrites branching out from the cell body, allowing them to integrate and process information before relaying signals. Efferent neurons transmit commands from the central nervous system to muscles and glands. A typical efferent neuron has several dendrites and a long axon that may branch into collaterals, ending in axon terminals where signals are transmitted to target cells. Structurally, neurons are classified as pseudo-unipolar, bipolar, anaxonic, and multipolar. Pseudo-unipolar neurons have a single axonal process, with the dendrite fused to the axon during development. Bipolar neurons have two relatively equal extensions from the cell body. Anaxonic neurons, found in the central nervous system, lack a distinct axon. Multipolar neurons, which are common in the central nervous system, have multiple dendrites and a highly branched structure, allowing extensive communication with other neurons. Nerves are composed of axons bundled together with connective tissue, allowing communication between different parts of the body. These bundles can be classified into sensory nerves, which carry information toward the central nervous system, motor nerves, which transmit signals to muscles and glands, and mixed nerves, which contain both sensory and motor fibers. The cell body of a neuron serves as its control center, housing the nucleus and organelles necessary for maintaining cellular function. Dendrites extend from the cell body and receive incoming signals from other neurons, playing a critical role in processing information. Axons, on the other hand, carry outgoing signals, transmitting nerve impulses to target cells. A key region of the axon is the axon hillock, where the action potential is initiated. This electrical signal travels along the axon, allowing neurons to communicate effectively with other neurons, muscles, or glands. This rapid transmission of impulses is essential for sensory perception, motor control, and overall nervous system function. A neuron is composed of several key structures that allow it to receive, process, and transmit signals. Dendrites extend from the cell body and serve as the primary receptors for incoming signals from other neurons. The cell body, also known as the soma, contains the nucleus and is responsible for integrating incoming signals. Once processed, signals travel to the axon hillock, where an action potential is generated. This electrical impulse propagates along the axon, a long extension covered by the myelin sheath, which increases the speed of signal transmission. The axon carries the impulse to the presynaptic axon terminal, where it reaches the synapse, the communication point between neurons. The synaptic cleft separates the presynaptic neuron from the postsynaptic dendrite of the receiving neuron. At the synapse, neurotransmitters are released, allowing signals to transfer to the next neuron, muscle, or gland. This flow of information, beginning with an input signal, leading to integration within the neuron, and culminating in an output signal, is fundamental to nervous system function. This image shows a multipolar efferent neuron, a type of neuron responsible for transmitting signals from the central nervous system to muscles and glands. Multipolar neurons are characterized by a large cell body with multiple dendrites extending outward, allowing them to receive input from many sources. The axon, which is not fully visible in this image, carries outgoing signals toward target tissues, facilitating movement and autonomic responses. Efferent neurons play a crucial role in motor control, enabling voluntary movements as well as involuntary functions like heart rate and digestion. The darkly stained nucleus within the cell body houses genetic material essential for neuronal function and maintenance. Surrounding structures, including smaller glial cells, provide support and protection for the neuron. This concludes our discussion on multipolar efferent neurons, a fundamental component of the nervous system. Fast axonal transport is a critical process that moves proteins and organelles between the cell body and the axon terminal, ensuring efficient communication and cellular function. This process begins in the rough endoplasmic reticulum, where peptides are synthesized and packaged by the Golgi apparatus. These newly formed vesicles and mitochondria are transported along the axon via microtubule networks, a process known as fast axonal transport. Once these vesicles reach the axon terminal, their contents are released into the synaptic cleft through exocytosis, allowing neurotransmitters to transmit signals to the next neuron. Following release, some vesicles undergo synaptic vesicle recycling, where membrane components are repurposed for future use. Additionally, retrograde fast axonal transport moves old cellular components back to the cell body, where they are broken down and digested by lysosomes. This bidirectional transport system is essential for maintaining the health and function of neurons, ensuring that synaptic communication remains efficient. This concludes our discussion on fast axonal transport. A synapse is the region where an axon terminal meets its target cell, enabling communication between neurons or between a neuron and another type of cell. The presynaptic cell is the neuron that sends the signal, while the postsynaptic cell is the one that receives it. These two cells are separated by the synaptic cleft, a small gap where neurotransmitters or electrical signals are transmitted. Synapses can be classified as either chemical or electrical. Chemical synapses use neurotransmitters to relay signals across the synaptic cleft. This allows for flexibility in signal modulation, but it also introduces a slight delay. Electrical synapses, on the other hand, involve direct ion flow through gap junctions, allowing for faster communication but with less control over signal strength and processing. Understanding how synapses function is essential to grasp how neurons communicate and how the nervous system regulates responses. This concludes our discussion on synaptic signaling. Glial cells play a crucial role in supporting and maintaining the function of neurons in both the central and peripheral nervous systems. Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system wrap around axons to form insulating myelin sheaths, which increase the speed of electrical signal transmission. Between these myelinated sections are nodes of Ranvier, small gaps that allow for rapid signal conduction. Another type of glial cell, satellite cells, are found in the peripheral nervous system. These cells, which do not produce myelin, provide structural and metabolic support to neurons in ganglia, clusters of nerve cell bodies located outside the central nervous system. In contrast, the nucleus of a neuron is found inside the central nervous system. Within the central nervous system, astrocytes have multiple subtypes and serve various functions, including maintaining the blood-brain barrier and regulating neurotransmitter levels. Microglia act as the immune cells of the brain, identifying and removing harmful substances to protect neural tissue. Ependymal cells line the ventricles of the brain and the central canal of the spinal cord, assisting in the production and circulation of cerebrospinal fluid. Together, these glial cells ensure that neurons function efficiently, providing structural support, insulation, immune defense, and fluid regulation. This concludes our discussion on glial cells and their roles in the nervous system. Glial cells provide essential support for neurons in the central nervous system, ensuring efficient communication and overall brain function. This figure highlights key glial cell types within the brain and spinal cord. Astrocytes are star-shaped cells that play multiple roles, including maintaining the blood-brain barrier, regulating ion concentrations, and providing structural support. Microglia function as the immune cells of the central nervous system, removing debris and protecting against pathogens. Ependymal cells line the ventricles of the brain and the central canal of the spinal cord, assisting in the production and circulation of cerebrospinal fluid. Another critical glial cell type is the oligodendrocyte, which forms myelin sheaths around axons in the central nervous system. These myelin sheaths enhance the speed and efficiency of nerve impulse transmission. The nodes of Ranvier, visible between sections of myelin, allow for rapid signal conduction. This complex network of glial cells ensures that neurons are well supported, protected, and capable of functioning efficiently. This concludes our discussion on glial cells of the central nervous system. Glial cells provide essential support and maintenance for neurons in both the central nervous system and the peripheral nervous system. This figure highlights the different types of glial cells and their specialized functions. In the central nervous system, ependymal cells create barriers between compartments and serve as a source of neural stem cells. Astrocytes regulate the environment by absorbing potassium, water, and neurotransmitters, while also forming the blood-brain barrier to protect the brain. Microglia act as immune scavenger cells, removing debris and defending against infections. Oligodendrocytes form myelin sheaths, which insulate axons and enhance the speed of nerve impulse transmission. In the peripheral nervous system, Schwann cells also produce myelin sheaths, ensuring efficient nerve conduction. Satellite cells support cell bodies and provide structural stability. Both Schwann and satellite cells secrete neurotrophic factors, which promote neuronal growth and survival. Together, these glial cells play a crucial role in maintaining homeostasis, protecting neurons, and facilitating efficient neural communication. This concludes our discussion on glial cells and their functions. In the peripheral nervous system, Schwann cells are responsible for forming myelin sheaths around axons, allowing for efficient nerve signal transmission. Each Schwann cell wraps around a small 1 to 1.5 millimeter segment of a single axon, leaving small unmyelinated gaps called nodes of Ranvier. These nodes enable rapid signal conduction by allowing electrical impulses to jump between myelinated segments, a process known as saltatory conduction. The formation of myelin begins as a Schwann cell wraps itself around an axon. As it continues to rotate, layers of cell membrane compact around the axon, forming the myelin sheath. This multi-layered insulation increases the speed and efficiency of nerve impulses, ensuring smooth communication between neurons and target tissues. Myelination is essential for the proper function of the nervous system, allowing for faster, more efficient signal transmission and reducing energy consumption in neurons. This concludes our discussion on myelin formation in the peripheral nervous system. Neurons are highly specialized cells, and their ability to regenerate is limited. If the cell body of a neuron dies, the entire neuron is lost, as the cell body contains the nucleus and essential organelles for survival. However, if only the axon is severed, the cell body and its attached segment may survive, while the detached portion degenerates. The consequences of axonal damage depend on the type of neuron affected. If a motor neuron is damaged, its target muscle may experience permanent paralysis due to the loss of nerve signaling. If a sensory neuron is damaged, the individual may experience a loss of sensation in the affected area. Regeneration is more likely in the peripheral nervous system, where Schwann cells help guide axon regrowth. However, in the central nervous system, regeneration is significantly less likely due to inhibitory factors in the environment and the lack of supportive glial cell activity. Neuronal repair is a complex process that occurs primarily in the peripheral nervous system, where Schwann cells facilitate axonal regeneration. When a neuron is damaged, the first step in repair is the leakage of axoplasm, which seals the damaged end of the axon. The attached segment of the axon swells as materials continue to move through axonal transport. Near the injury site, Schwann cells secrete neurotrophic factors, which help keep the cell body alive and stimulate the regrowth of the axon. However, the distal portion of the axon, which is no longer connected to the cell body, degenerates in a process called Wallerian degeneration. To support regeneration, macrophages, which are phagocytic immune cells, clear away cellular debris and secrete chemical signals that promote Schwann cell mitosis. These newly formed Schwann cells align to create a tunnel, guiding the regenerating axon back toward its target structure, such as a muscle or sensory receptor. This repair process allows for some functional recovery in the peripheral nervous system, though regeneration is often limited in the central nervous system. This concludes our discussion on neuronal repair mechanisms. When a peripheral neuron is injured, its ability to regenerate depends on the location and severity of the damage. If the axon is severed, the portion attached to the cell body may survive, while the distal segment, the part disconnected from the cell body, begins to disintegrate. This process is known as Wallerian degeneration. Following injury, the proximal axon swells, and phagocytic cells, such as macrophages, clear away debris from the damaged axon and myelin. This cleanup process is essential for preparing the injury site for possible regeneration. In some cases, the proximal axon can regrow through the remaining Schwann cell sheet, guiding it back toward its original target structure. If successful, the axon may reestablish a functional synapse, restoring some degree of neural communication. However, this process is more effective in the peripheral nervous system than in the central nervous system, where regeneration is far more limited. This concludes our discussion on peripheral neuron injury and repair. The resting membrane potential of a neuron is approximately -70 millivolts, and it is primarily determined by the cell's resting permeability to potassium ions. This electrical potential exists because the plasma membrane is more permeable to potassium ions than to sodium ions under resting conditions. Any changes in the potassium ion concentration gradient, such as in low potassium levels, known as hypokalemia, or high potassium levels, known as hyperkalemia, can alter the resting membrane potential. Similarly, changes in the permeability of the plasma membrane to either sodium ions or potassium ions will also affect the membrane potential, leading to changes in the cell's electrical state. These movements of charged particles across the membrane are essential for the generation of electrical signals, allowing neurons to transmit information throughout the body. This concludes our discussion on ion movement and the creation of electrical signals. This graph illustrates the changes in membrane potential over time, showing how neurons generate electrical signals. The resting membrane potential is the baseline electrical charge inside the cell, measured at approximately -70 millivolts. When a neuron receives a stimulus, the membrane potential depolarizes, meaning the inside of the cell becomes less negative as sodium ions enter. This upward shift in the graph represents the depolarization phase, where the neuron moves toward a more positive charge. If the depolarization reaches a certain threshold, it can trigger an action potential, allowing the signal to propagate along the neuron. Following depolarization, the neuron returns to its resting state, but in some cases, it may hyperpolarize, meaning the membrane potential becomes even more negative than the resting state. This occurs due to the efflux of potassium ions or influx of chloride ions, making it temporarily more difficult for the neuron to fire another action potential. These fluctuations in membrane potential are essential for neuronal communication, ensuring that electrical signals are transmitted efficiently within the nervous system. This concludes our discussion on the graph of membrane potential changes. Ion channels play a crucial role in regulating neuronal activity by controlling the movement of charged particles across the cell membrane. These channels are named according to the primary ion that passes through them, such as sodium channels, potassium channels, and others. The movement of these ions is essential for maintaining the resting membrane potential and generating electrical signals in neurons. Gated ion channels help regulate ion permeability, determining when and how ions enter or exit the cell. There are three main types of gated channels. Mechanically gated channels respond to physical forces, such as pressure or stretch. Chemically gated channels open in response to neurotransmitters or other signaling molecules. Voltage-gated channels open or close based on changes in membrane potential, allowing for the rapid transmission of electrical signals. By controlling ion flow, these gated channels enable neurons to generate and propagate action potentials, ensuring communication throughout the nervous system. This concludes our discussion on gated ion channels and their role in neuronal permeability. Graded potentials occur in the dendrites or cell body of a neuron. These signals vary in strength, meaning they can be weak or strong, depending on the stimulus. Graded potentials are used for short-distance communication within a neuron and typically do not travel far from their point of origin. If a graded potential is strong enough, it can trigger an action potential. Action potentials are initiated at the axon hillock and involve a large, brief depolarization of the membrane potential. Unlike graded potentials, action potentials are all or nothing responses, meaning they either occur at full strength or not at all. These electrical signals allow for rapid communication over long distances, enabling neurons to transmit messages efficiently throughout the nervous system. Together, these electrical signals ensure effective neural communication, allowing the body to process and respond to stimuli. This concludes our discussion on the two basic types of electrical signals. Graded potentials are changes in the membrane potential that travel a short distance within a neuron. Unlike action potentials, graded potentials may lose strength as they move through the cell. This loss occurs due to current leak, primarily caused by potassium ion leak channels, which allow ions to diffuse out of the cell, reducing the signal strength. If a graded potential is strong enough, it can reach the axon hillock, where it may trigger an action potential. To initiate an action potential, the membrane potential must reach a specific threshold voltage, allowing the neuron to fire and transmit a signal over long distances. Graded potentials can be either excitatory or inhibitory. Excitatory postsynaptic potentials bring the membrane potential closer to threshold, increasing the likelihood of an action potential. Inhibitory postsynaptic potentials, on the other hand, make the membrane potential more negative, decreasing the likelihood of an action potential. By regulating neuronal activity, graded potentials play a crucial role in information processing within the nervous system. This concludes our discussion on graded potentials and their role in stimulus strength. This figure illustrates the nature of graded potentials and how they spread within a neuron. Graded potentials are localized changes in membrane potential that occur when a stimulus causes ion channels to open, allowing ions such as sodium to enter the cell. At the stimulus point of origin, a graded potential begins with a strong amplitude, meaning a significant change in membrane voltage. As the signal spreads outward from this point, it gradually decreases in strength due to current leak. This occurs because potassium ion leak channels allow some charge to dissipate preventing the signal from traveling long distances. The graph in the figure shows how the amplitude of a graded potential decreases over distance. If the graded potential is strong enough and reaches the axon hillock with sufficient voltage, it may trigger an action potential, allowing the neuron to send a signal further. However, if the graded potential is too weak, it will fade away without initiating further communication. Graded potentials play a key role in neuronal processing, helping integrate incoming stimuli and determining whether a neuron will generate an action potential. This concludes our discussion on graded potentials and their characteristics. This figure illustrates the difference between subthreshold and suprathreshold graded potentials and how they influence the initiation of an action potential. A subthreshold graded potential begins above the threshold level at its origin, but gradually decreases in strength as it travels through the cell body. Due to current leak and resistance, the graded potential diminishes before reaching the trigger zone at the axon hillock. If the membrane potential at this zone is below threshold, the neuron does not generate an action potential, meaning the signal fails to propagate further. In contrast, a suprathreshold graded potential starts with a stronger stimulus at the same point on the cell body. Although it also decreases as it travels, it remains above the threshold when it reaches the trigger zone. Because the membrane potential at this location is above threshold, the neuron initiates an action potential, which then propagates along the axon to communicate with other neurons or target cells. This difference between subthreshold and suprathreshold graded potentials determines whether a neuron remains inactive or fires an action potential, playing a crucial role in neural processing and communication. This concludes our discussion on graded potentials and their effects on action potential initiation. Action potentials are rapid electrical signals that allow neurons to communicate over long distances. This process, known as conduction, involves the high-speed movement of an action potential along the length of an axon. Unlike graded potentials, action potentials follow the all or none principle. This means that either an action potential is generated at full strength or it does not occur at all. Once initiated, an action potential will propagate without losing strength, ensuring that signals reach their intended targets with accuracy and speed. This mechanism is essential for neural communication, enabling the brain and nervous system to coordinate movement, process sensory information, and regulate bodily functions. This concludes our discussion on how action potentials travel long distances. The conduction of an action potential along an axon is similar to a series of falling dominoes. Just as each domino falls in sequence, different sections of the axon membrane experience different phases of the action potential. This allows the electrical signal to propagate efficiently without losing strength. The action potential begins at the trigger zone of the neuron. As it moves along the axon, each segment undergoes the depolarization and repolarization cycle, ensuring that the signal is transmitted continuously. The graph in the figure shows recordings from different points along the axon, demonstrating that each region is in a different phase of the action potential at any given moment. This wave of electrical current allows neurons to transmit signals over long distances, ensuring rapid communication between the brain, spinal cord, and body. The reliability of this conduction process is critical for nervous system function, allowing for movement, sensation, and reflex responses. This concludes our discussion on action potential conduction and how neurons efficiently propagate electrical signals. An action potential begins when a graded potential reaches the trigger zone of a neuron and depolarizes the membrane to threshold voltage. This initiates a rapid sequence of ion movements that generate the electrical signal. During the rising phase of the action potential, voltage-gated sodium channels open, allowing sodium ions to rush into the cell. This influx of positive charge depolarizes the membrane, making the inside of the neuron more positive. As the membrane potential reaches its peak, sodium channels close, and voltage-gated potassium channels open more slowly. This marks the falling phase of the action potential, during which potassium ions exit the cell, repolarizing the membrane back to a negative voltage. If too many potassium ions leave, the neuron becomes temporarily hyperpolarized, making it even more negative than its resting state. Finally, potassium channels close, and the cell gradually returns to its resting membrane potential of negative 70 millivolts. This cycle of depolarization and repolarization ensures that neurons can generate and transmit electrical signals efficiently. This concludes our discussion on how sodium and potassium ions move across the membrane during action potentials. This figure illustrates the sequence of events that occur during an action potential, showing how changes in ion permeability create electrical signals within a neuron. The process begins at the resting membrane potential, where the inside of the neuron maintains a charge of approximately negative 70 millivolts. A depolarizing stimulus then occurs, triggering the membrane to depolarize to threshold. Once this threshold is reached, voltage-gated sodium channels open, allowing sodium ions to rush into the cell, making the inside of the neuron more positive. This marks the rising phase of the action potential. At the peak of depolarization, sodium channels close, and voltage-gated potassium channels open more slowly. This allows potassium ions to exit the cell, restoring a more negative membrane potential in the falling phase of the action potential. In some cases, the neuron hyperpolarizes, becoming even more negative than its resting state due to the continued efflux of potassium ions. As potassium channels close, the neuron returns to its resting membrane potential, preparing for the next action potential. The accompanying graph highlights these phases, showing fluctuations in membrane voltage and ion permeability over time. This cycle of depolarization and repolarization enables neurons to transmit electrical signals rapidly and efficiently, ensuring proper communication within the nervous system. This concludes our discussion on the phases of the action potential. This figure illustrates the function of voltage-gated sodium channels, which play a critical role in generating and propagating action potentials in neurons. These channels have two distinct gates: an activation gate, which opens rapidly, and an inactivation gate, which closes more slowly. At the resting membrane potential, the activation gate is closed, preventing sodium ions from entering the cell. When a depolarizing stimulus arrives, the activation gate opens, allowing sodium ions to flow into the neuron. This influx of positive charge further depolarizes the membrane, contributing to the rising phase of the action potential. As depolarization continues, the inactivation gate slowly closes, stopping the entry of sodium ions. This ensures that the action potential progresses in one direction and prevents continuous excitation. During repolarization, when potassium ions leave the cell, both gates return to their original positions, resetting the channel for the next action potential. The coordinated opening and closing of these gates enable neurons to rapidly send electrical signals, allowing for efficient communication within the nervous system. This concludes our discussion on the voltage-gated sodium channel and its role in action potential conduction. Voltage-gated sodium channels have two gates: an activation gate, which opens rapidly during depolarization, and an inactivation gate, which closes more slowly to stop sodium entry. This gating mechanism is essential for controlling the timing and direction of action potentials. During the absolute refractory period, action potentials cannot be generated because sodium channels are resetting. This ensures that signals move in one direction and prevents backward conduction. Regardless of stimulus strength, there is a 1 to 2 millisecond delay between action potentials. Following the absolute refractory period, the relative refractory period occurs, during which another action potential can be generated, but only with a stronger than usual stimulus. This period helps regulate the frequency of nerve impulses, preventing excessive firing and ensuring controlled signal transmission. Together, these refractory periods limit the rate at which neurons can fire, ensuring that signals remain coordinated and precise within the nervous system. This concludes our discussion on sodium channel gating and refractory periods. The refractory period is a crucial aspect of neuronal excitability, determining when a neuron can fire again after an action potential. It is divided into two phases: the absolute refractory period and the relative refractory period. During the absolute refractory period, voltage-gated sodium channels are either fully open or inactivated, making it impossible for another action potential to occur, regardless of stimulus intensity. This phase ensures that action potentials remain unidirectional, preventing the signal from traveling backward along the axon. Following this, the relative refractory period occurs as potassium channels remain open, allowing hyperpolarization of the membrane. At this stage, the neuron can fire again, but only if the stimulus is exceptionally strong, as the membrane potential is below resting levels. This dynamic interplay of ion channel states, membrane potential shifts, and refractory phases plays a fundamental role in signal conduction fidelity and neuronal communication precision. Without these periods, the nervous system would lack the ability to regulate action potential frequency and direction, leading to erratic and dysfunctional signaling. Understanding the temporal characteristics of the refractory period is essential in neurophysiology, particularly in fields such as neurology, neuropharmacology, and electrophysiology, where interventions in neuronal excitability can have profound clinical implications. Action potentials are conducted along the axon through a process known as depolarization. When a neuron reaches threshold potential, voltage-gated sodium channels open, allowing sodium ions to enter the cell, creating a wave of positive charge that spreads to adjacent sections of the membrane. This spread of charge is known as local current flow, and it plays a critical role in ensuring continuous propagation of the signal. As sodium channels open in a sequential manner, each new section of the membrane depolarizes, allowing the action potential to move forward without losing strength. To prevent backward conduction, sodium channels in the previously excited regions become inactivated during the refractory period, making them temporarily unable to reopen. This ensures that the signal moves in only one direction, from the axon hillock toward the synaptic terminal. This highly coordinated conduction mechanism is essential for rapid neural communication, allowing signals to travel over long distances efficiently and reliably within the nervous system. Local current flow is the key process that allows an action potential to propagate along the axon. When a section of the axon depolarizes, positively charged ions move toward adjacent sections of the cytoplasm, triggering the opening of voltage-gated sodium channels. This spread of charge initiates depolarization in the next segment of the axon, ensuring that the action potential moves in a forward direction. Meanwhile, on the extracellular surface, positive charges flow toward the depolarized region, completing the circuit of local current flow. This coordinated movement of charge is essential for continuous nerve signal transmission, ensuring efficient and rapid communication within the nervous system. The conduction of an action potential along an axon ensures rapid communication between neurons and their target cells. Once a graded potential reaches the trigger zone, it must surpass the threshold voltage to initiate an action potential. At this point, voltage-gated sodium channels open, allowing sodium ions to enter, rapidly depolarizing the membrane. The influx of positive charge spreads to adjacent sections of the axon, triggering local current flow and the activation of voltage-gated sodium channels further down. To prevent backward conduction, the refractory period follows each depolarization, ensuring that the signal moves in one direction. As potassium ions leave the cell, the membrane repolarizes, preparing for the next action potential. This precise sequence of depolarization, repolarization, and refractory period allows efficient and reliable signal transmission across neurons. The speed of an action potential depends on two key factors: neuron size and myelination. Larger neurons conduct action potentials faster due to reduced internal resistance to ion flow. However, the most significant factor in conduction speed is myelination. Myelinated neurons exhibit saltatory conduction, where action potentials jump between nodes of Ranvier, minimizing ion leakage and increasing efficiency. In demyelinating diseases like multiple sclerosis and Guillain-Barré syndrome, the loss of myelin slows or disrupts action potential conduction, leading to neurological impairments. Efficient signal transmission is crucial for coordinated movement, sensation, and cognitive function, making myelin an essential component of a healthy nervous system. Saltatory conduction is the rapid transmission of action potentials along myelinated axons. Unlike continuous conduction, action potentials leap from one node of Ranvier to the next, skipping the insulated regions covered by myelin. This process significantly increases conduction speed and reduces energy expenditure, as fewer ion channels need to be activated. However, in demyelinating diseases such as multiple sclerosis, myelin deteriorates, leading to current leakage, slower conduction, and even action potential failure. Efficient saltatory conduction is critical for rapid neural signaling, ensuring smooth movement, sensory processing, and reflexes. The conduction of action potentials is influenced by various chemicals that bind to ion channels, modifying their activity. Changes in extracellular ion concentrations also play a crucial role in neuronal excitability. For example, potassium and calcium ions are key regulators. Hyperkalemia, or elevated potassium levels, brings neurons closer to threshold, making them more excitable. Hypokalemia, or low potassium levels, moves neurons further from threshold, reducing their ability to fire action potentials. These chemical shifts can profoundly affect muscle contractions, nerve signaling, and overall neural function. Potassium is the primary ion responsible for the resting membrane potential. Under normal conditions, plasma potassium levels range between 3.5 to 5 millimolar, maintaining neuronal excitability within a balanced threshold. However, deviations in potassium concentration can alter neuronal excitability. In normokalemia, a subthreshold stimulus does not fire an action potential, while a suprathreshold stimulus successfully triggers one. In hyperkalemia, increased potassium levels bring the membrane potential closer to threshold, making neurons more excitable. A weaker than normal stimulus can now trigger an action potential. In hypokalemia, decreased potassium levels hyperpolarize the membrane, making neurons less likely to fire, even when exposed to a stimulus that would normally be suprethreshold. These shifts in potassium homeostasis play a critical role in nerve signaling, muscle contraction, and overall cellular function. Neurons communicate at synapses, which are specialized junctions where signals are transmitted between cells. Electrical synapses pass electrical signals directly through gap junctions, allowing for rapid and synchronized communication between neurons. Chemical synapses use neurotransmitters that cross the synaptic cleft and bind to receptors on the postsynaptic membrane, enabling more complex and modifiable signaling. Neurocrines are chemicals released by neurons that facilitate communication between cells. Neurotransmitters are a type of neurocrine that transmits signals across synapses, allowing neurons to communicate with other neurons, muscles, or glands. Acetylcholine binds to cholinergic receptors, which are classified into two types. Nicotinic receptors are found on skeletal muscles and in the autonomic divisions of the peripheral nervous system and central nervous system. These receptors function as monovalent cation channels, allowing the passage of sodium and potassium ions. Muscarinic receptors are located in the central nervous system and on target cells of the autonomic parasympathetic division of the peripheral nervous system. Amines are active in the central nervous system and are derived from a single amino acid. Serotonin is synthesized from tryptophan, histamine from histidine, and the catecholamines, dopamine, norepinephrine, and epinephrine, are derived from tyrosine. Noradrenergic neurons secrete norepinephrine, while adrenergic receptors bind both norepinephrine and epinephrine. These receptors are classified into alpha and beta types. Gamma-aminobutyric acid is an inhibitory neurotransmitter in the brain. It hyperpolarizes target cells by opening chloride ion gates, reducing the likelihood of an action potential. Neurotransmitters are released from synaptic vesicles located in the axon terminal of the presynaptic neuron. Their synthesis can occur in both the cell body and the axon terminal. The process of neurotransmitter release follows a classical pathway. It begins when an action potential arrives at the axon terminal, triggering the opening of voltage-gated calcium channels in response to depolarization. Calcium ions then bind to regulatory proteins, initiating exocytosis. Once released, the neurotransmitter diffuses across the synaptic cleft and binds to receptors on the postsynaptic cell, ultimately initiating a cellular response. Synaptic communication begins when an action potential reaches the axon terminal, causing depolarization of the membrane. This depolarization triggers the opening of voltage-gated calcium channels, allowing calcium ions to enter the presynaptic neuron. The influx of calcium binds to regulatory proteins, initiating the process of exocytosis. Synaptic vesicles containing neurotransmitters then fuse with the presynaptic membrane, releasing their contents into the synaptic cleft. Once in the synaptic cleft, neurotransmitters diffuse across the gap and bind to specific receptors on the postsynaptic cell membrane. This binding can lead to either excitatory or inhibitory effects, depending on the type of neurotransmitter and receptor involved. The binding event causes ion channels on the postsynaptic membrane to open or close, altering the membrane potential and influencing the likelihood of generating a new action potential in the postsynaptic cell. After neurotransmitters have delivered their signal, they are either broken down by enzymes, reabsorbed into the presynaptic neuron through reuptake transporters, or diffuse away from the synaptic cleft to terminate the signal. This tightly regulated process ensures precise control over neuronal communication, allowing for coordinated responses in the nervous system. Neurotransmitter activity must be terminated to prevent prolonged or excessive signaling in the nervous system. One mechanism of termination is diffusion, where neurotransmitters passively drift away from the synaptic cleft, reducing their concentration and effect on the postsynaptic receptor. Another method involves enzymatic breakdown, in which specialized enzymes degrade neurotransmitters into inactive components. For example, acetylcholinesterase rapidly hydrolyzes acetylcholine into acetate and choline, ensuring that synaptic signaling remains brief and controlled. Additionally, neurotransmitters can be removed through reuptake into cells. This process involves specialized transporter proteins that actively retrieve neurotransmitters from the synaptic cleft and transport them back into the presynaptic axon terminal for recycling or repackaging into vesicles. Some neurotransmitters can also be taken up by glial cells, which help regulate neurotransmitter levels and maintain synaptic function. These termination mechanisms collectively ensure precise control over neurotransmission, allowing for efficient communication between neurons while preventing excessive or prolonged activation of neural circuits. Neurotransmitter termination is a crucial process that ensures the proper regulation of neural communication. Once a neurotransmitter has transmitted its signal across the synaptic cleft, its action must be halted to prevent continuous stimulation of the postsynaptic cell. This termination occurs through three primary mechanisms: reuptake, enzymatic degradation, and diffusion. Reuptake involves the active transport of neurotransmitters back into the axon terminal of the presynaptic neuron for recycling or repackaging into synaptic vesicles. In some cases, neurotransmitters may also be transported into nearby glial cells, which help regulate their concentration in the extracellular environment. Another mechanism, enzymatic degradation, occurs when specific enzymes break down neurotransmitters into inactive metabolites. For example, the enzyme acetylcholinesterase hydrolyzes acetylcholine into choline and acetate, ensuring that the signal does not persist. Lastly, neurotransmitters can diffuse away from the synaptic cleft, reducing their local concentration and diminishing their effect on postsynaptic receptors. These termination mechanisms work together to maintain precise control over neural signaling, preventing overstimulation and allowing neurons to reset for future communication. The synthesis and recycling of acetylcholine play a vital role in neurotransmission, particularly in the peripheral and central nervous systems. Acetylcholine is synthesized in the axon terminal of the presynaptic neuron from two key components, choline and acetyl coenzyme A. The enzyme choline acetyltransferase catalyzes this reaction, producing acetylcholine, which is then packaged into synaptic vesicles for release. When an action potential reaches the axon terminal, voltage-gated calcium channels open, allowing calcium ions to enter the neuron. This influx of calcium triggers the fusion of synaptic vesicles with the presynaptic membrane, releasing acetylcholine into the synaptic cleft. Acetylcholine then binds to cholinergic receptors on the postsynaptic membrane, initiating a cellular response. To prevent prolonged stimulation, acetylcholine is rapidly broken down in the synaptic cleft by the enzyme acetylcholinesterase. This enzymatic breakdown produces choline and acetate. While acetate diffuses away, choline is actively transported back into the presynaptic neuron through sodium-dependent cotransport. Once inside the axon terminal, the recycled choline is used to synthesize new acetylcholine molecules, allowing the cycle to continue efficiently. This process ensures that acetylcholine-mediated signaling remains precise and controlled, preventing overstimulation while maintaining neurotransmitter availability for subsequent synaptic transmissions. The strength of a stimulus is encoded by the frequency of action potential firing in a neuron. A weak stimulus generates a graded potential that, if strong enough, reaches the trigger zone and initiates a low-frequency train of action potentials. This limited firing results in the release of a small amount of neurotransmitter at the axon terminal. In contrast, a stronger stimulus produces a larger graded potential, which leads to a higher frequency of action potentials. Since neurotransmitter release is directly proportional to the frequency of action potentials, a stronger stimulus results in a greater release of neurotransmitters into the synaptic cleft. This increased neurotransmitter release enhances the likelihood of activating the postsynaptic neuron or target cell, thereby amplifying the signal transmission. This frequency coding mechanism allows the nervous system to distinguish between weak and strong stimuli, ensuring an appropriate physiological response to varying sensory inputs. Neural communication is not always a simple one-on-one interaction between neurons. Instead, neural pathways often exhibit patterns of divergence and convergence. Divergence occurs when a single neuron sends signals to multiple downstream neurons, allowing for widespread distribution of information and coordinated responses across multiple targets. This is especially important in processes such as motor control and sensory perception, where a single signal needs to be relayed to various regions. Conversely, convergence happens when multiple presynaptic neurons synapse onto a single postsynaptic neuron, integrating signals from different sources. This convergence allows the nervous system to process complex information, such as summing up multiple sensory inputs to generate an appropriate response. Through these mechanisms, neural networks achieve higher-order processing, ensuring efficient and adaptive responses to stimuli. A divergent pathway occurs when a single presynaptic neuron branches out to influence multiple postsynaptic neurons. This type of signaling is crucial in processes where information needs to be distributed widely, such as in motor control, where a single motor neuron in the spinal cord can activate multiple muscle fibers to coordinate movement. It is also important in sensory systems, where a single sensory neuron can relay information to various regions of the brain for processing and response. In contrast, a convergent pathway involves multiple presynaptic neurons synapsing onto a single postsynaptic neuron. This type of neural integration allows the nervous system to compile signals from various sources before generating a response. For example, in the visual system, multiple photoreceptor cells in the retina send information to a single ganglion cell, refining and integrating the input before it is transmitted to the brain. Convergent pathways are essential for decision-making, allowing the nervous system to weigh multiple inputs before executing an action or response. Synapses on a cell body. The cell body of a somatic motor neuron is nearly covered with synapses from other neurons, allowing extensive communication and integration of neural signals. Axon terminals of presynaptic neurons make contact with the dendrites and soma of the postsynaptic neuron, ensuring that incoming signals can be processed and relayed efficiently. These multiple synaptic connections are essential for motor control, sensory processing, and complex decision-making in neural circuits. Purkinje cells. Located in the cerebellum, Purkinje cells are characterized by their highly branched dendritic trees, which enable them to receive thousands of synaptic inputs from other neurons. These cells serve as a prime example of a convergent pathway, where numerous presynaptic neurons provide input to a single Purkinje cell. The extensive dendritic arborization allows Purkinje cells to integrate and refine signals, playing a critical role in motor coordination, balance, and fine-tuned movements. Their outputs help modulate motor commands sent from the cerebellum to other parts of the brain and spinal cord, ensuring smooth and precise control of voluntary movements. Graded potentials are small, localized changes in the membrane potential that occur in response to synaptic input. These changes can either be excitatory or inhibitory, influencing whether a nerve impulse will be triggered. An excitatory postsynaptic potential causes depolarization, making the neuron more likely to send a nerve impulse. In contrast, an inhibitory postsynaptic potential causes hyperpolarization, reducing the likelihood of a nerve impulse being generated. A single graded potential is not sufficient to generate a nerve impulse. However, if multiple graded potentials combine through summation, either by occurring at the same time or in quick succession, they can reach the threshold at the axon hillock. When multiple signals from different synapses arrive at the same time, this is called spatial summation. When repeated signals from a single synapse arrive close together in time, this is called temporal summation. If the sum potential reaches the required threshold, channels in the membrane open, leading to the generation of a nerve impulse that travels along the axon. Neural pathways process and integrate information from multiple sources through different summation mechanisms. Spatial summation occurs when two or more neurons send signals to the same target neuron at the same time. These combined signals can have an additive effect, increasing the likelihood that the target neuron will generate a nerve impulse if the summed signals reach the threshold. Temporal summation, on the other hand, occurs when a single neuron sends multiple signals in rapid succession. If these signals overlap in time, their combined effect can bring the membrane potential to the required threshold, triggering a nerve impulse. Both spatial and temporal summation allow the nervous system to integrate and respond to multiple stimuli efficiently, ensuring appropriate neural communication and processing. Temporal summation occurs when multiple signals from the same presynaptic neuron arrive at the target neuron in close succession. If these signals, or graded potentials, are too far apart in time, they will not add together, and the neuron will not reach the required threshold to fire an action potential. This is known as no summation, where each signal remains below the threshold and fails to generate a nerve impulse. However, when multiple signals arrive closely together, their effects can accumulate or summate, leading to a larger overall depolarization. If this summation reaches the threshold at the trigger zone of the neuron, it will result in the generation of an action potential. This process allows neurons to integrate and process information over time, ensuring that only sufficiently strong or repetitive stimuli lead to nerve signal transmission. Spatial summation occurs when multiple presynaptic neurons send signals to a single postsynaptic neuron simultaneously. These inputs combine at the dendrites and cell body, allowing the neuron to integrate information from multiple sources. If each individual input is sub-threshold, meaning it does not reach the necessary level to trigger an action potential on its own, their combined effect may still be sufficient to generate an action potential when they summate at the trigger zone. In spatial summation, excitatory and inhibitory signals interact, determining whether the neuron reaches the threshold to fire. Excitatory synapses promote depolarization, while inhibitory synapses work against it by hyperpolarizing the membrane. If the sum of excitatory signals is strong enough to surpass the threshold, an action potential is generated, allowing the neuron to transmit information. This process enables the nervous system to integrate complex signals and modulate responses based on multiple simultaneous inputs. Synaptic inhibition occurs when an inhibitory signal counteracts excitatory inputs, preventing the postsynaptic neuron from reaching the threshold required to generate an action potential. In this process, an inhibitory neuron releases neurotransmitters that cause hyperpolarization of the postsynaptic membrane, making it more difficult for the neuron to reach the activation threshold. In this example, two excitatory neurons fire, attempting to depolarize the postsynaptic neuron and bring it closer to the threshold for an action potential. However, simultaneously, an inhibitory neuron also fires, generating an inhibitory signal that reduces the overall depolarization. As a result, the combined input of the excitatory and inhibitory signals does not reach the threshold, and no action potential is generated. This mechanism ensures precise control of neural signaling, preventing excessive excitation and allowing the nervous system to fine-tune responses. This summary provides an overview of key concepts related to the nervous system. It covers the organization of the nervous system, including the central and peripheral divisions, followed by a discussion on the different types of cells that make up the nervous system, such as neurons and glial cells. The section on electrical signals in neurons explains the generation and conduction of action potentials, highlighting the role of ion channels and membrane potentials. The module on cell-to-cell communication focuses on synaptic transmission, neurotransmitter release, and signal propagation between neurons. Finally, the integration of neural information transfer emphasizes how signals from multiple neurons interact through summation and inhibition, ensuring proper neural responses and regulation of bodily functions.